Plant hormones. Plant growth and development involve the integration of many environmental and endogenous signals that, together with the intrinsic genetic program, determine plant form. Fundamental to this process are several growth regulators, collectively called the plant hormones or phytohormones. According to a standard definition, plant hormones are small organic compounds synthesized by specific plant cells or tissues, active in low concentration, and promote or inhibit growth and developmental processes. The definition of a hormone used in animal physiology does not apply to plant hormones, although, like animal hormones, plant hormones are naturally occurring organic substances that profoundly influence physiological processes at low concentration. The site of synthesis and mode of transport for plant hormones, however, is not always so clearly localized. Although some tissues may be characterized by higher hormone levels than others, synthesis of plant hormones appears to be much more diffuse and not always localized to discrete organ or tissue. Plant hormones are able to exert their action locally or at a distance. Some are transported from one organ to another to organ to produce their physiological effect, and some others bring about changes in the same tissue or within the same cell where they are synthesized. These characteristics have led to consider that transport is not an essential property of a plant hormone. Growth in plants is defined as irreversible increase in size or volume. This is mainly driven by turgor pressure. During this process, cells increase in volume and become highly vacuolate. Growth also can be measured in terms of change in living biomass over a particular period of time. However, the living biomass of plants fluctuates in response to changes in water status, so this criterion may be poor indicator of actual growth. In these situations, measurement of dry weight are often more appropriate. The cell number is commonly used to measure the growth of unicellular organisms such as green alga chlamydomonas. In multicellular plants, however, cell number can be a misleading growth measurement because cells can divide without increasing in volume. Types of plant hormones. The concept of plant hormones originate from a classical experiment on phototropism, the bending of plants toward light carried out by Charles Darwin and his son Francis in 1880. The Darwins studied the bending of canary grass Phalaris canariensis coleoptile in response to unidirectional light. They demonstrated that a signal produced at the shoot apex travels downward and causes differential cell elongation in the lower parts of the coleoptile that resulted in it bending toward light source. This signal was subsequently shown to be IAA, the first known plant hormone. A small number of plant hormones have been shown to influence the plant growth and development. Tremendous progress has been made in the identification of plant hormones, discovery of their effects on plants, and elucidation of their chemical structures. For some of the plant hormones, considerable knowledge about their biosynthetic pathways have been obtained. Based on function or chemical structure, there are five major groups of plant hormones. These groups are auxins, gibberellins, cytokinins, abscisic acid, ethylene. In addition, there is a variety of other plant hormones including the brasinosteroids, polyamines, jasmonic acid, salicylic acids, and others. Despite their diverse chemical structures, most of the plant hormones are derived from three main types of metabolic precursors: amino acids, isoprenoid compounds, and lipids. The amino acid tryptophan and methionine serve as a precursor for IAA and ethylene, respectively. The isoprenoid compounds give rise to five classes of plant hormones: cytokinins, brasinosteroids, gibberellins, abscisic acid, and stigolactones. And finally, jasmonic acid is synthesized from a lipid precursor. Amino acid tryptophan give IAA, methionine give ethylene. Isoprenoid compounds IPP give cytokinins, gibberellin, abscisic acid, stigolactones, and brasinosteroids. Lipids alpha linolenic acid gives jasmonic acid.